Moving onwards to the small intestine. So remember we had that sphincter separating the stomach at the pylorus from the small intestine. And so now we're going to get into the small intestine. So this is a coiled hollow tube as well. It's about 8 to 10 feet long. It's between the stomach and the large intestine. And it is the primary site for digestion and absorption, right? Really important, that and absorption part. This is where we begin to see the movement of those micronutrients across the intestinal wall and into the underlying blood vessels. It's got 3 divisions. It's got the duodenum, which is the shortest proximal part, which is coming right from the pylorus of the stomach. So this is the duodenum here. Then it's got the jejunum and the ileum. So the jejunum is usually in the upper left corner here. The ileum is usually in the lower right corner here. And these are the 2 distal parts of the small intestine. From the ileum, we then go to the large intestine, okay? Functions of the duodenum. So the duodenum receives secretions from several different glands here. It's receiving pancreatic juice from the pancreas that has digestive enzymes in it. It also has bicarbonate-rich fluid, which helps to neutralize the acidity of that chyme, again, so that we don't start breaking down or digesting our own intestines. So we need that bicarbonate to neutralize the acidity of the contents coming through. And then we also see the secretion of bile into the duodenum. So bile, which is coming from the liver, can be stored in the gallbladder, and it's really important in the digestion of fats. So breaking down those fats by a process that we'll talk about called emulsification, and so that we can move them across the epithelial layer and into the underlying structures. Okay. So absorption, most of it happens in the small intestine. It's completed within the first 20% of the entire length of the small intestine. And the intestines are anatomically arranged to have a really large surface area for that absorption. So another adaptation that we see for absorption to occur. So it's kind of coiled and kind of jam-packed, as we've seen in the lab, into the central part of the abdomen there. And that alone helps to increase the surface area. But on top of that, inside the epithelial lining, it's very highly folded. So it's highly folded with structures that are called villi that I'll talk about. And then even more folded are the microvilli, which I'll also talk about. And so if we were to stretch the entire intestines out, we would have the surface area, like the square footage of the size of a tennis court, which is really, really large. I'm sure you would have heard that analogy before, that if you were to take your intestines, stretch them out, square footage, it would cover the size of a tennis court. So that highly folded nature on top of the coiled nature of the tube structures is what really improves that surface area for absorption. So the small intestines has these structures that are called villi, and villi, again, are the foldings that increase the surface area. Within the villi, we have blood vessels and lacteals. Blood vessels are going to be for the absorption of other nutrients outside of fat, so things like glucose and amino acids. The lacteal is a communication to the lymphatic system, and this is how we absorb fats. So fats are not absorbed into your blood, right? That would be catastrophic. We want fats to go into the lymphatic system. They can be sent to the liver, and then they can be sorted and processed appropriately, okay? On top of the villi, we have microvilli. So microvilli are another way to further increase that surface area by having these apical modifications of the epithelial cells that forms this brush border. Okay, can anyone remember the cells that we talked about before that also have a brush border formed by microvilli? Just a quick connection here. So what cells have we encountered before that have a microvilli that forms this brush border? Excellent, the proximal tubular epithelial cells, awesome, perfect. So the proximal tubular epithelial cells of the tubular structures of the kidneys also has this exact same adaptation, which aids in increased absorption, okay? Now, because of these out pouches or these the folded nature of the villi, we create these invaginations that are called the crypts of Lieberkühn. And they're similar to the gastric pits in that they're just kind of dipped down into the wall here. But these have different cells and different functions. So the epithelial cells of the crypts here help to secrete bicarbonate-rich fluid. This is how we neutralize that chyme that's coming from the stomach. And it's going to secrete that in the proximal layer of the intestine, so essentially in the duodenum, but it's then going to absorb that in the distal part of the intestine, which is the ileum. So we essentially secrete this bicarbonate-rich fluid out in the duodenum to neutralize that chyme. And then when we get down to the ileum, we reabsorb that bicarbonate-rich fluid to kind of clear the tract of that fluid and to allow for the beginning of the consolidation of the waste that moves into the colon. All right. Okay, just to re-emphasize here the connection between the villi, the microvilli, and the crypts of Lieberkühn. So essentially, this is the wall of our small intestine. So here's that cut surface of the tube itself. If we were to look closer at that tube, we would see these villi, which are these foldings of the epithelia here. And this is what I mean by the epithelia at different parts. So the question that someone asked earlier, which is an excellent question, how do these structures change at different parts? So as we'll see in the stomach, we saw the epithelia do a different thing with those gastric pits and glands. Here we're seeing the epithelia, same epithelia, but it's doing a different thing, right? It's creating a different structure. And that's what I mean by the modifications of the structures are different, but they're the same layers throughout different parts of the GI. So here the foldings create these villi, and then on top of the villi, as we can see on this closer view, we have the apical modifications of these cells, which are called the microvilli. Okay, both of these structures are serving to increase the surface area. Now, because of these out pouches of the villi, we have these invaginations here, which are called the crypts of Lieberkühn. This is where we have the secretion of that bicarbonate-rich fluid. Now, within a villus, so villus would be a singular structure here, we see the blood vessels and the lacteal. This is how we absorb those nutrients. So the amino acids that we've just broken down, so we start breaking those proteins down in the stomach from proteins to amino acid. We start breaking the starch down really in the mouth, but then even more so in the stomach and the small intestines. We break that down to glucose, and those things will move across this villus into the blood vessels here, right? The fats on the other end are going to be broken down into triglycerides, and they're going to be moved across, again, this villus, but into the green structure, which is the lacteal, right? And so that's how we can absorb those smaller nutrients into our blood for circulation.